Hi, my name is Aaron Calpo, alongside with Julian and Zhang, and we'll be covering the vestibular system. This is KIN 636, Spring 2021. So what is the vestibular system? It is the system processes for motor response of self-motion, head position, and spatial orientation in relation to gravity. This in turn helps stabilize gaze, head, and posture. The inner ear structure helps with guidance in any way where it reports motion and position of the head. This system is also a key component in postural reflexes and eye movements that work together with visual and proprioceptive systems. The vestibular labyrinth is the main structure of the vestibular system. In a broad sense, it uses hair cells to transduce physical motion into neural impulses. This is from the effects of gravity in which it coincides with translational and rotational movements of the head. This is located deep within the temporal bone and has an intimate relationship with the cochlea in which they are actually joined. More importantly, it contains two main components in which I will go into more detail. The first one is the Otolith organs, and the second would be the semicircular canals. Overall work hand in hand to localize the head. The purpose of the otolithic organs are to detect tilting and translational movements. The anatomy of the otolithic organs include the macula and the otolithic membrane. The macula contains the hair cells and supporting cells that detect motion, and this shift generates a displacement in the hair bundles, in which it causes a receptor potential in the hair cells. The way that they are oriented is that there is an axis of mirror symmetry so that the hairs on the opposite side of the striatum can be excited and the others will be inhibited. The sacral macula and the utricular macula work together to see where the head is oriented. The sacral macula is oriented vertically, in which it responds to vertical translations and or head tilts up or down, while the utricular macula is horizontal, in which it responds to translational movements and head tilts left or right. In terms of the otolithic membrane, it's a gelatinous layer above the macula. The otolithic membrane includes the otoconia, which has a sac of crystals of calcium carbonate. This makes it heavy and therefore any shift can be sensed if moved due to gravity. This allows the head to know which way is down. A notable fact is that the orientation on one side of the head is mirrored on the other side. As we will learn soon enough, the otolithic organs cannot distinguish between head tilts and translational movements, but is an overall blanketed movement. The difference can only be spotted by both the otolithic system and the semicircular canals working together. In terms of how the otolithic organs detect movements, it is that the hair bundle displacement will occur in response to translational head movements due to the mass in the otolithic membrane. This is reflected in response to the vestibular nerve that innervate otolithic organs. Prior to the tilt, the axon has a high firing rate and increases or decreases depending on the amount of tilt. When the head is level, the firing level of the neurons will return to base level. But when there is a tilt, the firing action potentials will remain until the head is back to normal. When the head comes from a tilt, it will lead to the decreasing firing rate until it returns back to neutral head position. The way that the hair is oriented in our hair bundles allow linear forces to be detected in any direction. The ultracell covers the horizontal plane or the transverse plane, while the sacral covers the vertical or the coronal frontal plane. With their orientations combined, it allows senses within 3 dimensions of the head. The variations of hair cell polarity and the otolith organs produce patterns of the vestibular nerve fiber that encodes head position. The semicircular canals sense head rotations and static head tilts. They can also sense external rotation forces as well. For example, a loop on a roller coaster. There are a total of 3 semicircular canals per ear, in which they have a bulbous expansion at their bases. Part of the canals is the crista, which contains hair cells and houses sensory epithelium. In addition, there is also something called the cupula, in which it bridges the ampulla and forms a viscous barrier where the endolymph can circulate. The semicircular canals work in that when the head tilts, the inertia of the endolymph produces a force to cross the cupula and causes a displacement between the hair bundles. The hair bundles in the crista are organized so that they are pointed the same way, whereas the saccular and utricular macula, which are part of the otolithic organs mentioned earlier, are dispersed in different directions. The formatting of the hair bundles within the semicircular canals lead to hair uniformly following the same direction the action of the head is. When the hair follows the same direction of the action, a polarization occurs. In turn, the action is opposite of where the head is facing. There is a hyperpolarization that occurs. This brings up the idea of pairings. These pairings occur between the left and right side of the head so that orientation can be made known by way of the organs polarizing or hyperpolarizing. In total, there are 3 pairs between the left and right side of the head. The first pair comes from the right and left horizontal canals. The other 2 remaining pairs come from the anterior canal on each side working with the opposite posterior canal. So the left anterior canal working with the right posterior canal and the right anterior canal working with the left posterior canal. These pairings indicate a couple of things. When one side of the pair starts to fire, the firing rate of the other half of the pair changes, having a reverse effect on one another. So when one side depolarizes, the other side hyperpolarizes. Now to talk about how the body perceives rotation. The overall orientation of the canals make them sensitive to moments only on their plane, creating this push-pull arrangement. The net result of the hair bundles provides the information about the rotation of the head, in which Julian will continue the discussion.
Before we continue, I'll briefly go over the anatomy of the semicircular canals and the regions important for sensing head rotations. The structure of the canals allows them to detect angular acceleration that occurs during head rotations. At the base of each semicircular canal are bulbous expansions called ampulla. Sitting inside of the ampulla is a gelatinous mass known as the cupula, which bridges the entire width of the ampulla and acts as a barrier that endolymphatic fluid can't go through. The ampulla also houses sensory epithelium containing the hair cells called the crista. The hair bundles extend from the crista and into the cupula. The semicircular canals are only excited by rotations that accompany head tilts, while the otolith organs are excited by head tilts and translational movements of the head. When the head turns along the plane of one of the semicircular canals, the movement of the endolymphatic fluid distorts the cupula. What happens is that the inertia of the endolymph produces a force going across the cupula, causing it to descend away from the direction of movement, causing the displacement of hair bundles within the crista. Innervated inside the semicircular canals are vestibular fibers which exhibit high levels of spontaneous activity, resulting in their ability to transmit information by increasing or decreasing their firing rate and more effectively encoding head rotations. The response of fibers innervating the hair cells of the semicircular canal has been studied and recorded in an experiment. The axonal firing rate of monkey's vestibular nerve has been observed during head rotations from being spun in a chair in 3 different phases: a period of acceleration, constant velocity, and instant deceleration to a stop. Maximum firing rate corresponded with the period of acceleration when the cupula is deflected. Minimum firing rate corresponded with the period of deceleration when the cupid is deflected in the opposite direction. And during the constant velocity state, firing rates return to baseline as the cupula returns to undeflected state. This can occur while head is still turning as long as the angular velocity is still maintained, such as on a ship, airplane, or roller coaster, where prolonged acceleratory arcs occur. All of this is part of the cupula endolymph system dynamic, which has a way of smoothing the transduction of head accelerations into neural signals. The angular acceleration caused by the high-frequency rotational head movements creates a velocity signal. During acceleration, the deflection of the cupula causes the activity to increase rapidly. During constant angular velocity, or low-frequency rotational movements, the firing rate drops, returning it to baseline rate and returning the cupula to its undeflected state. During deceleration, the cupula deflects in the opposite direction, decreasing the unit firing rate. The central vestibular pathway has two features distinguishing them from pathways for sight, touch, and hearing. First, it being multisensory, which we'll get into more detail with later. Second, the neurons of the vestibular nuclei function as premotor neurons and give rise to ascending sensory projections, which are pathways carrying sensory information from the body to the spinal cord and to the brain, and providing a short latency sensory motor arc, which is a quicker reaction time because of quicker brain processing that can drive extremely rapid compensatory eye and head movements in response to vestibular stimulation. The organization of these circuits contribute to rapid automatic behaviors such as reflexive eye movements to stabilize gaze, rapid postural adjustments to maintain balance, and higher order processes for our sense of spatial orientation and self-motion. The vestibular end organs communicate with the targets in the brainstem and cerebellum, areas that process information for computing head motion and position. Communication to these centers occur via the vestibular branch of cranial nerve 8. The cell bodies of the vestibular nerve reside in the vestibular nerve ganglion, also known as the Scarpa's ganglion. The distal processes innervate the semicircular canals and the otolith organs. The central processes project via the vestibular portion of the vestibular cochlear nerve to the vestibular nuclei and directly to the cerebellum. Within the vestibular nuclei is a large amount of canal-otolith convergence, a feature that enables the encoding of head orientation and motion through the environment. Information here in the vestibular nuclei and cerebellum is used to distinguish head tilts from translational head movements. The vestibular system's central projections participate in two major classes of reflex. The first one is responsible for maintaining equilibrium and gaze during movement, for coordination of the head and eyes in order to keep a fixed gaze on objects during movement. This ability is authorized by the vestibular ocular reflex, the VOR, a mechanism for producing eye movement that counters head movement. There are connections from the ocular motor nucleus to the medial rectus of the left eye and from the abducens nucleus to the lateral rectus of the right eye. The abducens nerve, or cranial nerve 6, is responsible for supplying the lateral rectus muscle, one of the ocular muscles of the eye, which moves it laterally. The abducens nerve fibers supply the lateral rectus muscle of the same side of the head. Neurons from the abducens nucleus travel through the medial longitudinal fasciculus pathway to the ocular motor nucleus, where the synapses occur with neurons controlling the medial rectus muscles of the other eye, moving the eye inward. The result is the coordination of eye movement. The abducens nerve allows you to look laterally with your left eye, and then the medial longitudinal fasciculus pathway fibers cause your right eye to look medially. When the head experiences rotary acceleration to the left, this activity that's happening in the left horizontal canal excites neurons in the left vestibular nucleus, resulting in the compensatory eye movements to the right. Circuits move the eyes in the direction away from the horizontal canal. When the head rotates left, it increases activity in the left horizontal canal. Circuits move the eye to the right, and vice versa if the head rotates right. Excitatory input from the horizontal canal on one side causes eye movement to the opposite side. In a similar way, head movement in other planes activate the other semicircular canals, causing those corresponding eye movements. The vestibular system's second major reflex is responsible for maintaining posture. Posture of the head is mediated by the vestibulocervical reflex, the VCR, and the posture of the body is mediated by the vestibulospinal reflex, the VSR. In the VCR pathway, axons within the medial vestibular nucleus descend in the medial longitudinal fasciculus to reach the spinal cord. Stimulation in the semicircular canals in response to head rotations causes reflex activity in the neck muscles to regulate head position. The VSR pathways of the medial vestibular nuclei, which is demonstrated in this image, is projected bilaterally in the ventral horn's medial part and mediate head reflexes in response to activation of the canals. The ventral horn cells of the limb muscles become activated because they receive axons from the lateral vestibular nucleus via the lateral vestibular tract. The neurons in the nucleus receive input from the cerebellum, allowing this area of the brain to influence posture.
The vestibular system works with the visual system to allow us to still see objects clearly, even as our head is moving. It also helps us maintain awareness of position. On top of that, sensory receptors in the skin, joints, and muscles provide information to the brain on movement, on body parts and their relationship to each other, and also on body parts and their relationship to the environment. From this information, messages are sent from the brain to the muscles to tell it to move and make the necessary adjustments to body position that will maintain balance. As we walk in a translational movement, the vestibular system gets activated along with visual flow across the retina. The example in the book given is perceiving movement as you are stationary and watching a moving train. This perception of self-movement is also known as vection. The vestibular system works with the visual system. The cerebellum is a major target of ascending pathways and also provides descending information to the vestibular nuclei. The cerebellum and the vestibular system work closely with each other to be able to distinguish head tilts from translational movements and passive movements from self-generated movements. The parts of the cerebellum that are targets of the vestibular system include the flocculus, paraflocculus, nodulus, uvula, and rostral vestigial nucleus. The vestibulo-ocular reflex is very plastic, meaning that it is able to compensate for any damage to the vestibular periphery. Ivo Kohler conducted a series of experiments in the 1950s where he found that after wearing inverted prisms for a week, he could function well enough to ride a bike down a busy street. This is due to the flocculus and paraflocculus having Purkinje cells that send inhibitory signals to the vestibular nucleus and creates adaptations of the reflex. The nodulus and uvula have individual Purkinje cells that integrate signals that signal for either head tilt or translational movement. Predictive signals from the cerebellum cancels out vestibular and proprioceptive signals in the rostral vestigial neurons, which are generated from self-motion, meaning that these predictive signals from the cerebellum can distinguish active movements of the head from passive ones. The cerebellum This slide shows the pathways to the thalamus and the cortex, which are carrying vestibular information. The lateral and superior vestibular nuclei project to the thalamus. The lateral and superior nuclei also send axons to the ventral posterior nuclear complex of the thalamus, which then projects to two areas in the vestibular cortex. Three cortical areas are also the targets of these projections, one being just posterior to the primary somatosensory cortex, one to the area between the somatosensory cortex and the motor cortex, and one to the posterior parietal cortex. Electrophysiological studies of individual neurons show that cells in various cortical areas respond to proprioceptive, visual, and vestibular stimuli. When the head is rotated in the horizontal plane to one direction, the vestibular afferent nerves on that side fire at higher rates, while the afferents on the other side fire at slower rates. You can see that here in the picture where the person is rotating their head to the right, and the right afferents are firing at a much higher rate than the left side. Fluid rotates opposite of the rotation of the head, which is what causes this higher firing rate. A phenomenon known as physiological nystagmus then occurs, where the person's eyes will counter the rotation and move to the left slowly, and then quickly in the other direction to the right. This is also known as the vestibulo-ocular reflex. Spontaneous nystagmus occurs when there is damage to one side of the vestibular system, and therefore the brain believes that the difference in baseline firing and the damaged canal, which has no firing, is representative of head movement even when there is none. This is also known as vertigo. Vestibular stimulation can be used to determine whether someone, even if unconscious, has any damage to their brainstem. The patient is placed on their back and their head is elevated to 30 degrees above horizontal. This allows for the horizontal canals to be vertical. Flushing cold water through a normal patient's ear can mimic rotational head movement and thereby cause their eyes to slowly move toward the flushed ear, then quickly away. This is shown in the left column of the image. Abnormality due to being unconscious would result in the eyes only moving slowly towards the flushed ear. This is shown in the second from left column of the image. If there are any brainstem lesions affecting vestibular nuclei or their connections or peripheral nerves, there can be diminished or no response from the eyes. This is shown in these last two columns.